And the zona pellucida, it gets dissolved by an enzyme which is called as acrosin as soon as the sperm fertilizes. The number of nucleotides present in the DNA of a primary oocyte is approximately 3.3 billion base pairs. In situ conservation means conserving the organisms in its natural habitat itself, not taking it somewhere else and then conserving it. Hello everyone, welcome to this model question paper discussion session for 2nd PUC Biology. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So I have divided this session, that is the model question paper, into 2 sessions, wherein in this particular session, that is session 1 of this particular discussion, we will learn part A and part B of the question paper. So we will try solving the part A and part B, which consists of multiple choice question in question main 1 and in the main 2 fill in the blanks and 2 marks question in the main 3, that is in part B. So before we begin with solving with the question paper, we shall see what are the general instructions that you need to follow while writing the answers for the question paper. So this is what will be printed in the question paper. So the first one is the question paper consists part A, part B, part C, part D and part E. So you have to attend part A, part B, part C and part D. Part E is only for the visually challenged children. So next is part A. It consists of 2 divisions, that is main 1 and main 2. Same way, part D also consists of 2 divisions, that is main 5 and main 6. So part A, which consists of 2 divisions, main 1 and main 2, it carries 1 mark each, and part D, it carries 5 mark each. And all the parts are compulsory. There is no choice in questions between the parts. So all the parts are compulsory. Only under that part, in that a particular main, there will be options wherein you can eliminate few questions based on the number of options that they have given. But otherwise, all the parts are compulsory. And the answers for part A, this you have to keep in mind because it was introduced this year. So part A should be written in the first 2 pages of the answer booklet. So you can't skip or you can't strike it out and write it somewhere else. That will be considered invalid. It will not be corrected. So whatever you write the answers for part A, that is main 1 and main 2, in the first 2 pages of the answer booklet, only that will be considered for correction. So please have that in mind, because earlier there was an option where anywhere you would write it also the correction would be done, but that is not how it is for 2025. Next is part E. As I said, it contains questions for visually challenged students only, wherein 2 or 3 questions will be given specially for these kids. So moving further, let's look into the skeleton of the question paper. So as I said, part A consists of 2 mains, that is main 1 and main 2, wherein in main 1 we have MCQs wherein options will be given and we have to select the correct options, and where it should be in the first 2 pages itself of the answer booklet. Next is fill in the blanks, wherein earlier exactly if there was 5 questions in the fill in the blanks, 5 options would be given, but now they are adding an extra option which will be even more challenging for you because the options might look similar. So you have to be very cautious there. And next one is part C, that is 3rd main, wherein answer any 5. So out of the 8 questions that are given, any 5 needs to be answered. So in part C, that is 4th main, any 5 needs to be answered, and in part D, which consists of 5th main and 6th main, carrying a total of 5 marks each. Each question carries 5 marks. There any 4 should be answered and any 1 needs to be answered. So this is how the question paper pattern is. And the 6th main is entirely for visually challenged students. So moving further, let's try solving the question paper. So in this session, we'll try solving part A, that is main 1 and main 2, and part B, which consists of 2 marks questions. So under part A in the first main, that is select the correct alternative from the choices given below. So for each question, 4 choices, that is A, B, C and D will be given. We have to select the correct answer. So for the first question, they have given 2 statements here, statement 1 and statement 2. So we need to see whether both the statements are true, whether only one is true, the second one is false, or both are false. Like that we need to check. So statement 1 says that apomixis is the production of seeds from unfertilized ovule. That is true, right? Because that is what apomixis means. Statement 2 says that embryos produced from apomictic seeds are not generally identical to the parent plant. That is false, because your fertilization will not take place. Therefore, they have to be identical to the parent plant. So, therefore, the statement is false. So, they have given the options here. Statement 1 is true, statement 2 is false, statement 1 is false, statement 2 is true, statement 1 and statement 2 both are true, statement 1 and statement 2 both are false. So, we'll look into the answer. So, the answer is A, that is statement 1 is true and statement 2 is false. So, the explanation goes like this. Apomixis refers to the production of seeds from the unfertilized ovule. So, therefore, the statement is true. Now, embryos produced from the apomictic seeds, they are genetically identical to the parents because their pollens are not involved there. So, without fertilization process, that is from the ovule from the female and the pollen from the male, without that, or the male gamete from the male, without that the development of the seed takes place. So, therefore, they will be identical to the parent, genetically identical. So, therefore, the second statement was false. So, option A, statement 1 is true, statement 2 is false is the correct answer for this question. So, moving on to the next question. The question says choose the correct option from the table given below for the formation and dissolution of the parts that are labeled in the diagram. So, first you need to look into the diagram. So, what are the parts that they have labeled? They have labeled ovum or ova, and they have labeled zona pellucida. So, zona pellucida and ovum they have labeled. So, they have given here 2 columns formed by and dissolved. So, formed by, so ova is formed by. So, the first one is formed by, ovum is formed by, and zona pellucida when it is dissolved is what we need to see here. Zona pellucida layer. So, we have to choose the correct one here. So, ovum formed by primary oocyte. Zona pellucida dissolved. Is it dissolved before fertilization? Next is ovum formed by primary oocyte. Is zona pellucida dissolved after fertilization? Next is ovum formed by secondary oocyte, and is zona pellucida dissolved before fertilization? The same way, ovum is formed by secondary oocyte and zona pellucida, is it dissolved after fertilization? Now, let's look into the answer. So, therefore, ovum is formed by secondary oocyte. Ovum is formed by secondary oocyte and zona pellucida gets dissolved after fertilization. So, how we look into the explanation. So, first we'll look into what is the zona pellucida. So, zona pellucida is a layer that covers the ovum, and the zona pellucida is formed during the development of the secondary oocyte within the ovarian follicle. And the zona pellucida, it gets dissolved by an enzyme which is called as acrosin as soon as the sperm fertilizes, because once the fertilized zona pellucida is present, so that it allows only one sperm to fertilize the ova or the egg. Otherwise, all the sperms, millions of sperms that are traveling, can go and fertilize the egg. So, to prevent that, the zona pellucida is there. So, as soon as one sperm fertilizes the egg, the zona pellucida will get dissolved. So, therefore, zona pellucida dissolves after fertilization. So, therefore, after fertilization is the correct answer here. And next, the zona pellucida prevents other sperms from entering the oocyte, and I've already told about that. So, therefore, what did we get to know? Ovum is formed from the secondary oocyte. You know that chart, right? That is the chart that is there. So, therefore, in that we got to know that ova is formed from the secondary oocyte and zona pellucida dissolves soon after fertilization by the sperm. So, therefore, the correct option is D, ova, that is secondary oocyte after fertilization. Moving further. They have given few options here. So out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF result. So what is this IVF? In vitro fertilization, right? So in vitro fertilization, what is done? Under suitable conditions or simulated conditions in the laboratory, the ova from the donor and the sperm from the donor or the father, or the ova from the donor or the mother is brought and it is fertilized, and then it is put into the fallopian tube or to the uterus of the mother. So here they are asking the question, the correct stage for transfer of the fallopian tube for successful IVF results. So here they are planning to transfer the in vitro fertilized egg. They are planning to transfer it into the fallopian tube. So therefore, in what stage it will be transferred into the fallopian tube is the question above. So the answer is embryo up to 8 blastomere. So when the embryo reaches 8 blastomere, that is when they will transfer the embryo into the fallopian tube of the mother. From there, it will move and then it will attach itself in the endometrium. So that is how this happens. So the correct stage for embryo transfer in vitro fertilization is up to 8 blastomeres, because the embryos at this stage have higher chances for implantation. So that is why they will wait up till the embryo reaches 8 blastomere stage and then they transfer it. So the explanation is given here. So they have given the explanation what is meant by IVF and what is the optimal stage for transfer of embryo. So here they have given about an experiment related to T.H. Morgan, wherein 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye, normal body, white eye and miniature body is due to. So what is the reason for the Drosophila to have red eye, normal body, white eye and miniature body? So they have given the number here 37.2%. So what is the answer? Is it loosely linked and shorter distance between the genes? Is it tightly linked and shorter distance between the genes? Is it loosely linked and longer distance between the genes? Is it tightly linked and longer distance between the genes? So you have a small topic in the textbook that is for molecular genetics about linkage and recombination, if you remember, right? So the question is based on that. So the answer is loosely linked and longer distance between the genes. So 37.2% recombinant Drosophila progeny. Here recombination is happening between the Drosophila having red eye and normal body and white eye and miniature wing. So here what they said, 37.2% recombinant Drosophila progeny in T.H. Morgan's dihybrid cross experiment is due to loosely linked genes and longer distance between the genes. So if this is a chromosome that we're talking about, so here they will be loosely linked and the distance between the two also will be longer. It's not like they are present shorter. So this is how it is. So this is what they are talking about. So therefore, this is because the recombination frequency is higher, which indicates that the genes are farther apart. So here what happens now when there is recombination happening, say for example, there is a chromosome here and it is recombining with this, then the chances of recombination happening is more. Here what will happen when the recombination takes place or crossing of the chromosomal arms take place, both A and B will go to the other chromosomal arm. But here it is not like that. Only A will go because they are placed far apart. So that is why 37.2% recombination is because of loosely linked and longer distance between the genes. So moving further, the number of nucleotide pairs present in the DNA of the primary oocyte of a newborn in human. So what they have given here the numbers, they have given the length of the DNA strand. So is it or the number of obviously the number of nucleotides will give us the length of the DNA strand. So is it 3.3 into 10 to the power 9, 6.6 into 10 to the power 9, 13.2 into 10 to the power 9, 3.3 into 10 to the power 9. So it is The answer is option A, 3.3 into 10 to the power 9. So which means that the number of nucleotides present in the DNA of a primary oocyte is approximately 3.3 billion base pairs. Instead of writing 3.3 billion base pairs, we represent it by 3.3 into 10 to the power 9. So therefore, this is the answer. Moving further. The factors that affect Hardy-Weinberg equilibrium are given below. So this is a direct question. So there are a set of factors which are given in the textbook. Factors affecting Hardy-Weinberg equilibrium, right? So here they have given a number of factors. So which one is the correct one is what we need to choose. So first one is crossing over, independent assortment. The second option is crossing over, mutation. The third option is genetic drift, crossing over. The fourth option is independent assortment and mutation. So say for example, this is correct for this and the other one is wrong. You can't choose this as the correct option. All the points that they have given in the option should match with the limitations or with the factors that affect Hardy-Weinberg's principle. So the answer here is option B. That is 2, 3 and 4 is correct. So 2, 3 and 4 is correct means the factors that affect Hardy-Weinberg's equilibrium is crossing over and mutation, genetic drift and crossing over, independent assortment and mutation. So therefore that is the right answer here. So answer is B, 2, 3 and 4. So moving further. Interferons are most effective in making non-infected cells resistant against the spread of which of the following disease in humans. So the first thing you need to remember here is when exactly does alpha interferons or interferons get produced. So interferons are nothing but they are barriers. So you would have studied while studying about the human diseases. What are the barriers that are present? So we have certain physical barriers, physiological barriers, chemical barriers, cellular barriers and all that, right? So here it is a barrier and that barrier's name is interferon. So whenever a body gets infected by a virus, that is when the interferons will get activated or interferons will be produced so that the viruses will not spread to the neighboring healthy cells. So therefore, what did we get to know from this sentence that I just said now? So it is a viral disease. So here, which one is a viral disease? Is it AIDS, ringworm, ascariasis, amoebiasis? It is AIDS because AIDS is caused by human immunodeficiency virus, that is HIV. So therefore, always remember interferons are produced only during a viral infection. Therefore, option A is the correct answer here. Not ascariasis because ascariasis is nothing but it is produced by roundworm. And ringworm, it is produced by Trichophyton rubrum, which is a fungi. And amoebiasis is produced by Entamoeba histolytica, which is a protozoan. So therefore, the right answer is, so AIDS is the right answer because interferons are produced, are proteins that are produced as an immune response to the body whenever there is a virus attacking a body so that the virus cells do not spread to the neighboring healthy cells in a body. So therefore, answer is AIDS. So moving further. Next question, the human host cells in which the gametocytes of malaria parasites develop are. So we all know that in the life cycle of malaria, half of the life cycle happens in the host and the other half happens in the that is in the vector, that is inside the gut of the mosquito, right? So here the answer is when exactly is gametocytes produced? So, is it thrombocytes? Is it in the liver cells? Is it in the erythrocytes? Or is it in the leukocytes? So, the answer is erythrocytes. So, first they will attack the liver cells. From the liver cells, they will move into the RBCs. And in the RBC, which are also called as the erythrocytes, in that the male and the female gametocytes are produced. So, therefore, the answer is erythrocytes. So, the explanation is given here for that. So, moving further, which of the following water samples in the table given below will have a higher concentration of organic matter? So, it's something related to the biological oxygen demand. So, if we get to know that in the water sample, that is in the sewage water, if high amount of biological oxygen demand is there, there is demand for high amount of oxygen. Therefore, it is understood that the sewage is highly polluted or organic matter is present in that more and it's not yet been cleaned. So, therefore, the question is, which of the following water sample in the table given below will have a higher concentration of organic matter? So, water sample they have given here. So, is it because level of pollution is high and value of BOD is high? Of course, if the pollution in the water is high, the value of BOD will also be high. Is it because it's low and low? So, what is the question here? Will have a higher concentration of organic matter. So, if it is low and low, it doesn't mean it will have higher concentration of organic matter. No. So, this water sample is wrong. Next is low and high. No, because if the pollution is low, then the value of BOD will not be more. So, therefore, not this sample also. Next, if the pollution is high, the value of BOD will not be low. So, therefore, not this option. So, the correct answer is A, high and high. So, therefore, if pollution is high, then the BOD is also high. So, therefore, organic matter in the water sample A will be more because their pollution is also high and therefore, the biological oxygen demand is also high there. So, moving further, this is about the recombinant DNA technology wherein they have given the steps of the recombinant DNA technology. We have to arrange it in the right order. So, the first one they have given here, insertion of recombinant DNA into the host organism, amplification of the gene of interest using PCR. Next is cutting of DNA at specific location. Next is obtaining the foreign product. Next is downstream processing. Next is isolation of the DNA. So, the answer starts with isolation of the DNA. So, first what we need to do, we need to isolate the DNA. After isolating the DNA, we need to cut the strand that we need, right? So, therefore, the second one is cutting. After that, we have to, the cut strand should be increased in number. Clones of that needs to be created, which is amplification, that is replicas of that needs to be created. So, therefore, we have to use the PCR machine, that is the polymerase reaction, chain reaction technique to amplify or increase the number of copies of the cut DNA. After amplification, we have to introduce it into the foreign organism. After introducing into the foreign organism, then from the foreign organism, we have to obtain the foreign product. After that, we have to sell it to the market or do the downstream processing. So, therefore, the option D is the correct option here. So, option D is the right option. So, this is the explanation for that, wherein first one, extraction of the DNA from a source organism, then use certain restriction enzymes to cut the DNA at specific points of isolate the gene of our interest. After that, using polymerase chain reaction to multiply that copy of the DNA, then joining the cut DNA fragment into the vector, that is into the foreign organism, then introducing the recombinant DNA into a host organism, and then using a marker gene to identify recombinant DNA cells from non-recombinant DNA. So, all this is the explanation. So, moving further, DNA in a clone of cells followed by detection using autoradiography is called. So, how do we detect the DNA of our interest? By using autoradiography technique. So, we detect it by using a probe. So, we need a probe. So, DNA is a clone of cells followed by detection using autoradiography is called as probe. So, we introduce a probe. So, that probe can be identified in the autoradiography procedure. So, with that we will get to know that the DNA of our interest is present there and we can isolate that. So, the explanation for that is given here, wherein a probe is a fragment. So, first we need to know what a probe is. A probe is a fragment of DNA or RNA that is used to detect the complementary DNA sequence that is present in a clone of cells, because multiple clones will be there. Which one we need to detect that? We need to introduce a probe there. And the probe, how can we get to know that the probe has gone and attached to the complementary DNA strand? Why? Because the probe will be auto-radio labeled. So, therefore, when it goes and attaches to that particular DNA strand and when we see it under the UV light, it will emit light. So, therefore, we can understand that, okay, this is the DNA of our interest and we can isolate it. So, that is the explanation that is given here. So, next question. Jiva was growing a bacterial colony in a culture flask under ideal laboratory condition. So, what was he doing? So, in a laboratory, he was growing a bacterial colony. where the resources sooner or later becomes limiting. Which of the following equation will represent the growth curve of the bacteria? So we need to see what is the growth of the bacteria when the resources required for the growing bacteria becomes limiting or gets declined. So that is what the question is. So what is the equation we will look into? So the answer is dN by C, that is dN by dt is equal to Rn into K minus N divided by K. So the correct equation for bacterial growth under limiting resources is the logistic. It's not exponential, it is logistic, because after a certain time the growth of the bacteria will decline due to the limited availability of the resources. Therefore, it is a logistic growth equation. So therefore, the answer is dN by dt, that is option C. Next, moving further, which of the following food chains is the major conduit for the energy flow in terrestrial and aquatic ecosystem? So they've considered two aquatic ecosystems here, that is, one is terrestrial and the other one is aquatic ecosystem. So we need to see in both of these which is a food chain that actually circulates. So is it grazing and grazing? Is it detritus and detritus? Is it detritus and grazing? Is it grazing and detritus? So they have given separately two columns, terrestrial and aquatic. So both in terrestrial and aquatic, it is the grazing food chain that actually runs. So grazing and grazing, option A is the right answer, because in terrestrial ecosystem, the major source for energy flow is typically the grazing food chain, and similarly in an aquatic ecosystem also the major reason or the major role for energy flow is in the grazing food chain itself. So therefore grazing food chain is the right answer for both. So therefore option A is the right answer here. So the next question, the 14th question, which of the following is not an example? So keep this in mind. We are asking not an example of in situ conservation. So what do you mean by in situ conservation? In situ conservation means conserving the organisms in its natural habitat itself, not taking it somewhere else and then conserving it. So the right answer here is National Park and Zoological Parks, because it is in the National Park and Zoological Parks that the organisms will be conserved in their natural habitat. So that is why option B is the right answer here. So next question. Exploration of molecular, genetic and species level diversity for novel products of economic importance here. So what do we call exploration of molecular, genetic and species level diversity for novel products of economic importance? So what is that called? Is it called biofortification, bioprocessing, bioprospecting, biodiversity? So the answer will be bioprospecting. That is, bioprospecting is the process of studying the molecular and genetic diversity of species, determining their economic importance and making use of that. So it involves discovering commercialization of these products, novel products, and therefore it is a narrow utilitarian category. So therefore the answer is bioprospecting. So we'll move to the second main of part A, that is fill in the blanks by choosing the appropriate word or words from those given below. So they have already given the words in the bracket here. We need to fill in with the appropriate answers. The interstitial space in seminiferous tubule consists of immunologically competent cells. So we call it as competent cells. Next question, the version of biogenesis is accepted by majority. So this is an evolution related question. Biogenesis means formation of new life, and that was accepted by majority in the first form of life arose slowly through evolutionary process from non-living molecules. So these scientists or philosophers, they believe that new organisms evolved from non-living molecules. Next question. Filariasis pathogens are transmitted to a healthy person through vectors, right? So say for example, elephantiasis. So that is a filarial pathogen. So it is transported through a vector, that is through a mosquito. So therefore vectors is the answer. Next, Swiss cheese which large holes is produced from bacteria. So bacteria is the one that creates, because carbon dioxide will be released and therefore a lot of large holes will be created in the Swiss cheese because of this bacteria. That is Propionibacterium shermani, right? Next question. The host cells which have the ability to incorporate foreign DNA with them are called. So with them are called, they are called recombinant cells. It's not competent cells, but they are called recombinant cells. So the host cells which have the ability to incorporate, that is to recombine the foreign DNA. We call it as recombinant cells. So recombinant cells is the right answer here. Next, part B, that is third main, which is of 2 marks each. Here, complete the tabular column given below with respect to the male gametophyte of angiosperm. So, cells of the male gametophyte, they have given shape of the nucleus. So, cells of the male gametophyte. What are the cells of the male gametophyte? The larger one, that is the vegetative cell. And the smaller one, it is the generative cell. So when talking about the male gametophyte, we are talking about, they have mentioned here angiosperms. So therefore, we are talking about the pollen grain, right? So therefore, it will have 2 types of cell, that is cells of the male gametophyte, vegetative cell and generative cell. So what is the shape of the nucleus in the vegetative cell and generative cell is what is the question here about. So vegetative cell it is, large and irregular, and in generative cell it is spindle shape. So this is the right answer. That is cells of the male gametophyte, vegetative cell, generative cell. Shape of the nucleus of the cells irregular and spindle. Moving further, mention the 2 medical grounds on which the pregnancies are subjected to termination. So why medical termination of pregnancy is done? What are the conditions in which MTP is accepted is what we have to write. Wherein the continuing the pregnancy could be harmful or fatal to the mother. So if the child can be harmful or fatal, or continuing the pregnancy, if it causes problem to the mother, then it can be terminated. Or if the fetus has any incurable disease or genetic disorder, then with the will of the parents, then pregnancy can be terminated. Or if the child may be at risk of higher abnormalities, which may be difficult for it to lead a better life later on, so at that time it can be aborted. And other one is the mother may be concerned about passing off an inherited disease to the child. That is advice for medical termination of pregnancy. So therefore, these are the 4 points that you need to write. Next question, derive the phenotypic and genotypic ratio of a cross between AB blood group of parents. So between AB blood group of parents, so the genotypic and phenotypic ratio. So the phenotypic ratio for a cross between 2 AB blood group of parents, it is 1A is to 2AB and 1B. That is one child. There is possibility for one child having a blood group A, possibility for 2 children to have a blood group of AB, and possibility for one child to have a blood group of B. So this is the genotype. And next, this is the phenotype. So next is genotypic ratio, that is IAA, because for A it is IA. For B it is IB. So therefore here also the ratio, and for AB blood group it will be IA and IB. This is the phenotype. This one, and this is the genotype. So they are asking what is the possibility of the phenotype, ratio and genotype of individuals of parents having the AB blood group. So therefore the genotype is 1IA, because here it is 1A, therefore the genotype will be 1IA, 1IA. And here it is 2AB, it will be 2IA, IB. And here it is 1B, it will be 1IB and IB. So this is the answer for this question. Moving further, which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? So what do you mean by hnRNA? hnRNA is nothing but heterogeneous RNA, meaning they don't have the same kind of gene, because we know that in the DNA strand, that is on the mRNA strand in eukaryotes, it is a heterogeneous mRNA which consists of both exons and introns, right? And in the mRNA strand, when we say mRNA, so to put it like this, hnRNA, it will contain exons and it will contain introns. mRNA is a mature RNA, mature messenger RNA. So therefore, it will contain only exons, which are the coding sequences. So therefore, here which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? Which sequences? Exons are found, right? So therefore the answer that you need to write is the sequence of bases transcribed from DNA that are found in both heterogeneous, that is hnRNA, and messenger RNA are exons. That is 5' untranslated region, 3' untranslated region, start codon will be there, stop codons will be there. All these are the exons itself. So therefore, this is the answer that you need to write. So moving further, potato tubers and sweet potato tubers are the result of convergent evolution. Justify the statement. So potato tubers and sweet potato tubers are a result of convergent evolution because they are analogous organs, right? Where they are similar in function. What is their function? To store food. And they have evolved independently from different species, and they are also different to look out. Their origin is different. So therefore, this is the answer that you need to write. Moving further, what are biofertilizers? Mention its significance. So you have to write what are biofertilizers. If you write the definition of biofertilizer, you will get 1 mark. If you write 2 significance, which carries half a mark each, you will get 1 mark. So therefore, a total of 2 marks you will get here. So biofertilizers are substances that contain living microorganisms that helps the plants to grow better and give better yields. And they are renewable resources, they are eco-friendly, all that. So what are the significance? So significances, they help in storing the or restoring the nutrients of the soil, and they minimize the use of chemical fertilizers, and they will prevent soil pollution. All these are the uses. So any 2 if you write, you will get marks. So this answer definition carries 1 mark. Any 2 points you write, it will carry 1 mark. This is the answer for question number 26. So moving further, name any 4 recent extinct organism as per IUCN Red List, that is International Union for Conservation of Nature and Natural Resources. They are the ones that maintain a red data book or the red list. So in that, which are the extinct organisms? Any 4 that we need to mention. So dodo bird of Mauritius Island, then quagga of Africa, thylacine of Australia, Stella sea cow of Russia, and 3 subspecies of tiger, that is Bali, Javan and Caspian tiger. Any 4 if you write, each will carry quarter mark. So therefore, a total of 2 marks. So if you just write 2, you will get only 1 mark. 4 needs to be written there. So this was about this session wherein we learned the multiple choice questions. We tried solving the multiple choice question, the fill in the blanks and also the 2 mark question. So in the next session wherein we'll continue the other parts of the model question paper, which will include the 4th main and 5th main as well as the 6th main. That is the 3 marks and the 5 marks question. So this was about this session wherein we learned how to answer the MCQs and the 2 mark question and answers as well as the fill in the blanks. I hope the session was quite fruitful. So see you in the coming session discussing these parts of the question paper. So see you in the next session. Thank you.